Now, the mutation of the lac operon, we have studied that the mutation of the lac-I gene in such a way that the repressor encoded no longer will get binded to the lactose. In this case, the repressor would bind to the operator regardless of the presence or absence of the lactose, and the operon would never be transcribed at high levels. Then mutation of the lac-I gene in such a way that the repressor no longer binds to the operator. In this case, the operon would never be repressed. So, mutation of the lac-I gene it in such a way that the repressor will no longer bind to the operator gene. In this case, the operon would never be repressed and transcription would be carried out continuously. So, this is known as constitutive transcription. Then mutation in the operator region in such a way that the wild type repressor does not recognize it, that is, the repressor recognizes the specific DNA sequence of the operator region. In this case, there will be no binding of the repressor to the operator and transcription will go on continuously. So, in this manner, with the mutation of the lac operon, we have seen that how the transcription goes on continuously. Now, the catabolite repression. Expression of the lac operon can also be regulated in another way. Glucose is preferable to lactose as an energy. Glucose is preferable to lactose as an energy source. Hence, if glucose is present in the environment, the transcription is reduced or lac operon is down regulated. Transcription of the lac operon, it requires another protein called the catabolite activator protein, that is the CAP, or for short. This CAP protein, it binds to the lac promoter and enhances the transcription. So transcription of the lac operon, it requires some other protein called as the catabolite activator protein, CAP. So this CAP, it will CAP protein, it will bind to the lac promoter gene and it will enhance the or bring about the transcription. But it occurs only after CAP binds to the small molecule called cyclic AMP, small c AMP, that is adenosine monophosphate. So without CAP will not bind to the promoter and no transcription will occur. So to bring about the transcription, the in the lac operon, it requires the CAP, and the CAP, it should be binded along with the lac promoter gene to bring about the transcription. And for this CAP to get binded with the lac promoter gene, it also requires a small molecule called the cyclic AMP. So in the absence of CAP will not be able to bind to the promoter and no transcription will occur. So in the previous examples involving the lac operon, we can assume that CAMP was present and the CAP-CAMP complex was bound to the promoter gene. The CAMP is produced by an enzyme called adenyl cyclase. In the presence of glucose in the environment, adenyl cyclase is inhibited. So when the glucose is present in the medium, adenyl cyclase is inhibited and CAMP production drops. So adenyl cyclase is getting stopped and the CAMP production it will get drop down. So thus there is no CAMP to bind to CAP, and in this situation the CAP will not bind to the lac promoter. So in the presence of CAMP only the CAP will be able to bind and the transcription process will take place. But uh, if the glucose is present in the environment, then adenyl cyclase will get stopped secreting and the CMP production will get dropped. So in the absence of CMP, the CAP will not get binded to the lac promoter and so no lac transcription will take place. So in this way, the bacterium does not produce enzymes for lactose metabolism when they are not necessary because of the presence of glucose. Then beta galactosidase breaks lactose into glucose and galactose. When enough lactose has been metabolized, the glucose or one of the products accumulates and it causes the repression or stopping of the lac operon, or the stop of the transcription process in the protein synthesis will take place. Even when enough lactose has been metabolized, the glucose will get accumulated and it will cause the repression of the lac operon. Repression means stopping of the function of the lac operon, or transcription process will get stopped. Then merits of operon model in gene regulation. It is a very simple yet informative model of gene regulation in prokaryotes. It is a very well understood model of gene regulation in prokaryotes. This model is based on empirical results and has been studied on different prokaryotes. This model is of two types: inducible operon and repressible operon. Then now we'll study the next, that is the repressible operon. So these some of the four were the merits of the inducible operon model. Then the repressible operon, or the a protein molecule which prevents the transcription, is called repressor. That is, such type of a repressible operon is such type of a protein molecule which will stop the transcription, and so it is called as the repressor. And the process of inhibition of transcription is called repression. Repression. Then repressible operons are regulated by the end product of the metabolic pathway and not bioreactant in the metabolic pathway, such as the lactose in lac operon. An example of repressible operon is the tryptophan. This encodes the enzymes which are responsible for the synthesis of the amino acid tryptophan, that is TRP for short. The TRP operon is regulated by TRP, which is the product of the metabolic pathway. In tryptophan operon, the TRP repressor, it will only bind to the operator when tryptophan is present opposite to the lac repressor. The repressor binds to the tryptophan and undergoes a change in the shape or conformational change, and this change in shape, it allows it to bind to the operator gene and bring about the blocking of transcription process. So because tryptophan is needed for repression, it is referred to as a co-repressor in this system, as opposed to lactose being an inducer. So tryptophan operon is the repressor operon, we can say, model. So tryptophan repressor means it will bring about the stop in the transcription process, transcription mechanism of the in the protein synthesis. So because tryptophan is needed for repression, that is for stopping, it is referred to as a co-repressor. It is acting as a co-repressor in the system, as opposed to the lactose being an inducer. Whereas the lactose is the inducer. It will start the transcription process, and the tryptophan will act as a stopping molecule for the, or the tryptophan is acting as a repressor molecule, or repression will take place, or the stop of transcription process in the protein synthesis will be blocked, or it will be stopped. So when tryptophan is absent, the repressor will not bind to the operator and the transcription occurs. Thus, if there is plenty of tryptophan around and no more is needed, the transcription is blocked. If there is no tryptophan around, it needs to be synthesized and the transcription will occur. So in the same manner, just as we have seen in the presence of lactose and in the absence of lactose, so here also in the presence of uh, in the absence of tryptophan, we will see that it needs to be synthesized and transcription will occur. Whereas in the presence of tryptophan, the repressor will not bind to the operator and transcription will occur. And in the absence of tryptophan, the transcription uh, sorry, in the absence of tryptophan, the transcription will occur, and in the presence of tryptophan, the transcription will get blocked or stopped. In other words, it allows the production of the enzymes for tryptophan synthesis. Now this is the table showing the comparison of inducible and repressible operons. Inducing means starting and repressing means stopping. So now this is the inducible operon and these are the repressible operon. Example, lac operon, this is the tryptophan operon. The regulation, it is controlled by lactose, here it is end product is tryptophan. Addition of lactose, it will induce the or bring about the transcription in the repressible operon, no need uh, of the lactose. Absence of lactose, it will block the transcription. In the repressible operon, it is not seen. Addition of tryptophan, it will not work in the inducible operon, but it will work in the repressible operon, that is represses transcription process. Lack of tryptophan, that is the absence of tryptophan, will not be seen or it is not present in the inducible operon, but in the repressible operon it will enhance or bring about the transcription process. So binding of repressor with operator, transcription is blocked, and uh, here also the transcription is blocked when the repressor gets binded with the operator gene. No binding of repressor with the operator, then in the inducible operon transcription will occur, and in the repressible operon also transcription will occur. So repressible operons are organized in much the same way as inducible operons. There are structural genes under the control of a promoter and operator, and there is a gene encoding a repressor. The mutation will affect the gene regulation as follows. Mutation in the repressor gene in such a way that it no longer binds tryptophan. When repressor does not bind tryptophan, there will be no change in its structure, no conformational or change in shape will be seen, no change in its structure, and it will not get binded with the operator, and transcription will occur. Mutation in the repressor gene in such a way that it no longer binds the repressor molecule or repressor gene, and in such situation transcription will take place. Mutation in the operator in such a way that it no longer binds to the repressor, and in such situation also transcription will occur. Then uh, the mechanism of gene regulation we will study next class.